Hello everyone, welcome to the ITLS Academy. Today we are going to discuss the topic that is your detection of yeast and mold. Okay, so why it is important to detect the yeast and mold in a water sample? Because when we are detecting something, first of all we have to know that what is the importance of that experiment. Okay, we all know that these yeast and mold contaminate the water sample, and when these contaminated sample consumed by the consumer, it will harm the it will harm them. Okay, it also affect the uh, health of the human being, affect the health of the human being. This yeast and mold become a reason of the food spoilage. Okay. So ultimately, the main reason for the detection of the yeast and mold into a sample is that to preserve the health of the human being and even a person who are consuming it is the main reason. Okay, so that's why we are going to discuss how we detect these yeast and mold. Okay, and we all know that these yeast and mold are what they are come under the category of microorganism. That means that we cannot seen by it with our naked eye. That's why the tests are performed for their detection. So let's start the today's lecture. Introduction. Microorganisms which are capable of forming colonies in a selective medium, and here the medium that we are going to use is a chromophenicol glucose yeast extract agar medium at 25 degrees Celsius temperature. Okay, now here the selective medium means that when we use this media, that is your chloroamphenicol glucose yeast extract agar medium, then in that case it will only allow the growth of the and mold and suppress the growth of the other microorganism. That's why we are using the selective medium. That's the introduction part. Here we use a selective medium, and this selective medium suppress the growth of the other microorganism but only allow the growth of the yeast and mold. And this growth we will take it at 25 degrees Celsius, because yeast is the type of the fungus. So the fungus will grow at around 24 to 25 degrees Celsius temperature. That's why we're using it. Now the next is your now principle. Chromophenocol yeast glucose agar is what it is, a selective medium which recommended for a isolation and enumeration of the yeast and mold. Okay, selective medium means that it only allow the growth of the yeast and mold. Okay, that's why it is called a selective medium, and it is used for the isolation and the enumeration of the yeast and mold. This medium contain yeast extract, and this yeast extract provide a nitrogenous nutrients and vitamin B complex. Okay, so this is a composition of the CYGA media. Okay, so it contain a yeast extract. This yeast extract is used to provide the nitrogenous nutrient, and along with it also provide a vitamin B complex. Dextrose is used in the form of energy source, and chloro chloramphenicol, a thermostable antibiotic which suppress the bacterial flora. Okay, so this chloramphenicol, it's what it is, a thermostable antibiotic, and when we use it, it will suppress the other bacterial flora and only allow the growth of the yeast and mold. Aerobic incubation of plate is done at 25 degrees Celsius plus minus 10 degrees Celsius, and the colony will be count on third day, on fourth day, and on fifth day. One of that day we will count the colony, but the incubation temperature will be around 25 degree Celsius. Now the next is your culture media. So here we are using a chlorophenicol glucose yeast extract, and the composition of this CGYEA medium is what it contain: a yeast extract, yeast extract. Okay, so this composition is for a 1 liter. This composition is for a 1 liter. So in the amount of the yeast extract, we take 5 gram per liter. We also take a glucose. We also take a glucose, and the amount of the glucose is around 20 gram per liter. And chloro chloroamphenicol, we take it 0.1 gram per liter, and the last we take a agar 14.9 gram per liter. Okay, so this is composition for a 1 gram. If you want to prepare for it 500 liter or 100 liters, so you have to change this composition according to the amount of the distilled water. So this is about the culture media. Now the next is your procedure. Okay, so one more thing I just want to add here. Here the pH of this media should be around 6.6 plus minus 0.2. Okay, when we prepare this media, we have to maintain its pH around 6.6. Then what should we do? We autoclave it. We autoclave it. CGYEA. Okay, pH should be 6.6 plus minus 0.2. Then we autoclave it, and this autoclavation should be done at 121 degrees Celsius, 15 psi, and for 20 minutes. Okay, then after that what we have to do, we clean the laminar. Okay, and in hot air oven we sterilize the glassware around 150 degrees Celsius for 2 hours at least. Okay, then after that all the things what they are free from the microorganism, then we do a experiment. So here it is written then that aseptically clean the surface of the bottle, pouch, cup containing the sample with the help of the 70% of the ethanol and filter it 250 ml. What we have to do, we clean the the bottle at which the samples are there with the help of the 70% of the ethanol. Then we filter it, and we take a water sample through a membrane filter of we filter the water sample with the filtrate membrane. Pore size of that filter membrane is around 0.45 micrometer by using a sterile membrane filtration assembly. Now after that we place this filter paper into a CGYEA media. Now how we place it? Suppose that this is your petri plate. Now what we have to do, we pour the petri plate inside, we pour the media into the petri plate. We pour the media into the petri plate, and then after that we place that filter paper in just above. Okay, and leave it for a third day, fourth day, and for fifth day. But the temperature will be 25 degrees Celsius. Now we count a colony at third day, then at fourth day, and then at fifth day. Okay, then after that observation. Okay, so observation, we observe the colony of yeast and mold on third day, fourth day, and fifth day of the incubation. And it is also advisable that to examine the plate at the end of the 3 days for yeast colonies, which are smooth, moist, elevated, or surface colonies. Mold colonies are easily recognized with the help of the profuse growth of hyphae. Okay, then expression of the result. We all know that presence and absence of this yeast and mold are counted at, suppose that this is your plate. Okay, now we count it. This is your yeast and mold colony. So we count it like this: 1, 2, 3, 4, 5, 6, 7, 8. Now it is represented at as 7 colonies per 100 ml of sample. This is the representation of the result. Okay, so you just count the colonies and represent at in per ml. This is the expression of the result. So this is all about the determination of the yeast and mold. I hope you will understand, but if you have any query related to this topic, you can ask me in a comment box, or for a further detail you can follow the ITLS Academy on different platforms, that is Instagram, Facebook, Twitter, YouTube, WhatsApp and LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play, or you can also ask your query to the helpline number that are already mentioned in your slide, that is 7080833450, and visit our website www.itlsacademy.com. Thank you for watching. Please like, share and subscribe our channel.